and welcome to the Excel Cycle. Today we will be talking about Mendelian genetics and its three main laws. First, let's define a few terms that we will be encountering throughout the lesson. Let's begin with a gene. When we talk about a gene, we are referring to a specific sequence of nucleotides at a location of the chromosome. Individuals have two copies of each gene, one from their mother and one from their father. Genes are said to be expressed and produce what is called a phenotype. Phenotype is an outward expression of genes. For example, someone may have the phenotype of blue eyes, which was determined by their genes. Another individual may have a different sequence within this gene that gives them the phenotype of green eyes. These variations are called alleles. These alleles differ in their nucleotide sequence and produce different phenotypes as a result. Some alleles are said to be dominant over others. This means when an individual is heterozygous, or has two different copies of the same gene, the individual will take on the phenotype of the dominant allele. In this case, blue is dominant. Next, let's talk about Gregor Mendel and why he is so important. Mendel is referred to as the father of genetics and postulated three main laws. These were law of equal segregation, law of independent assortment, and law of dominance. Let's talk about Mendel's second law first, the law of independent assortment. This states that unlinked or distantly linked segregating gene pairs assort independently at meiosis 1. But what does this really mean? To simplify this, let's look at homologous pairs of chromosomes during metaphase 1 of meiosis. The green chromosomes represent the paternal chromosomes, and the purple show the maternal. In simple terms, the law of independent assortment means that these homologous pairs can arrange themselves in many ways. Let's look at the first pair of homologous chromosomes, with each parent providing alleles for the same gene. Let's pretend this gene is for eye color. These homologous chromosomes can arrange themselves just the way they are, but also in the opposite configuration. You can see that the alleles for gene 1 from each parent are able to assort themselves in different ways, like this, or like this. The other half of the law states that the arrangement of gene 1, or any gene for that matter, has no effect on the arrangement of the other genes. This means that gene 2 is able to arrange its alleles in either way, independently of what arrangement happened for gene 1. The same is true for gene 3, or any other gene. This means that alleles of different unlinked genes are able to assort independently, just as the law states. Now we can go over Mendel's first law, the law of equal segregation. This law states that only one allele for each gene in an organism is distributed to each gamete. Let's remember that meiosis 1 ends with the separation of homologous pairs of chromosomes, and meiosis 2 ends with the separation of sister chromatids. Let's again look at gene pair 1 and the alleles associated with it. At anaphase 1, these homologous chromosomes are separated and bring their corresponding alleles with them. This segregation of alleles of the same genes at anaphase 1 creates two daughter cells ready for meiosis 2. In meiosis 2, these sister chromatids will separate and produce two daughter cells each, for a total of 4 gametes. It's important to see that different alleles of the same gene are separated at anaphase 1, whereas sister chromatids containing the same alleles are separated at anaphase 2. Lastly, the law of dominance states that when an organism has alternate forms of a gene, or alleles, the dominant allele will be phenotypically expressed. Think back to our eye color example. Blue, or big A, is dominant over green, or little a, so in the presence of both alleles, blue is expressed. Thank you for following along with today's lesson. Be sure to check out our other videos, as well as comment what you would like to see in the future.